Here we go, the first lab for semester 2. This lab is called Cells and the Structure of Life, and I'm going to walk you through exactly everything you need to do. I suggest that you watch this video along with having another tab open that has the actual lab in it and a third tab or a different device open with the actual lab report. Then watch for a ways on the video, pause it, do that part of the lab, come back to the video, watch another segment, pause it, and then work through the lab slowly and carefully that way, because if you do that, I guarantee you're going to turn in a piece of work that's going to earn you the grade you want and you deserve. All right, let's get going. So this one, when you come down into the coursework, you're going to see 4.02, Cells and the Structure of Life. These are the lesson pages that you need to go through before you even begin the activity. Right here is the Google Doc that you can open up. There will be a link and it will force you to make a copy. Once you've made that copy, please come up to the share button and enter in my email address. If it doesn't automatically pop up, you're going to enter it in, wvs.klohoff@cesa9.org. Please share your work with me immediately. That way I can watch as you're working on it, especially if we're coming to a connect time or a support session, and give you pointers so that you aren't wasting your time and you're doing the work the right way right off the bat. All right, once you have made that copy, you can go back to the activity pages here, open up the lesson pages, and again, you're going to slowly and carefully work through the pages. I give you the suggested work list. There's a video each week that tells you what pages are suggested for you to look at each day. The assignment is on the last page of the lesson. And once you get there, I might need to do a little finagling with my screen here to get it to fit. All right, you might hear my cat in the background. For some reason, she's very, very angry. Up here, you're going to see a link to the lab report. You can do your report through this link. There's also a grading rubric. I have included the grading rubric at the bottom of the document. There it is right there. So if you want to know how I score your work, this is exactly the tool that I use to award points for each of the different areas. All right, so back in here, welcome to Cell Theory Headquarters. In today's investigation, you will complete 3 mini experiments to collect evidence about cell theory. Begin by choosing one component of cell theory to investigate. Then, continue your work until you have collected evidence from all 3 areas. First, we have all organisms are composed of cells. The second one, cells come from existing cells. And finally, cells are the basic unit of life. I'm going to click on all organisms are composed of cells. Activity 1. In this experiment, you will use a microscope to find evidence that all living things are made of cells. As you complete this activity, record your observations in your lab report. Okay, let's click the next button. Roll over to identify the parts of the compound microscope. You're going to read through, make sure you read through each of the plus signs. It forces you to look at all of them before the next button appears. So make sure you take your time to read through those parts of the compound microscope. Compare 3 types of living things under the microscope to see if they are all made of cells. You can select any of these items and bring it to the microscope to view. So we're going to look at a plant cell. We're going to pick it up and drag it over here and then select the high power, and we're going to get a visual here. Now it's very blurry, so we can use this tool here, there, to make it become clearer. These are plant cells from an onion. Record observations in your lab report, then select another slide and drag it to the microscope to view it. Here's the key to science. When you have to write an observation, you're literally going to describe what you see. So when I look at this slide of the onion plant cells, I see different colors. I see different shapes. I see things of different sizes. I'm noticing that there seem to be like these cells are in lines, and then there are other areas that are clumped. All of those things are what you would put in your observation. So come here to the lab report. The top part, you're going to put your name in here, your instructor's name, the date, the name of the lab. This lab is called Cells and Structure of Life. That's worth points. The objective, why are we doing this? What are we learning about? We're learning more about just cells and the structure of life. If you can't figure out why we're doing it at the beginning, then wait until you're done and see what kinds of things did they ask me about? What kind of questions did I have to answer? That's telling you that that's the objective. That's what the lab is there for you to learn. All right. Then here, you're going to list the function of all those different parts of the compound microscope. Remember, I told you to read them. And here we have our observations from each specimen under the microscope. So the plant cell, I would describe the color, the shapes, the sizes, anything else I notice. Obviously, I'm not giving you the answer. You have to describe this in your own words. All right, then we should be able to go. Why is it not letting me go? There we go. Right, then when I'm ready to move on to the next slide, which is an animal cell, I just go down here to get the hand shape and grab it. And then I can view the animal cell. I need to click on this to make it focus. Oh, that's not getting it. There we go. These are red blood cells from an animal. Record observations in your lab report, then select another slide and drag it to the microscope to view it. So again, I'm going to look at this and I see colors, I see shapes. I'm noticing things are much further apart than they were for the plant cell. There's a lot of empty space on this slide compared to the plant cell. These red blood cells, yeah, there's only a few on this example. And if I look back at that plant slide or the plant cell, everything is very tightly together. There's almost no empty space here. It's pretty solid. So I would write my descriptions again for what I'm seeing here in my lab report. So in the animal cells, I would put the color, the shape, sizes, other things I notice. List all of the things that you observe. That's what you're putting into that space. Then we have the bacteria cell. So coming in here, we put the bacteria cell in, turn on the microscope, hit our buttons to get the bacteria. Oh, look at that. These are bacteria cells. This shape of bacteria is called a rod shape. Record observations in your lab report, then select Next to move on to the self-check. Notice color, shape. We are told these are rod-shaped. I'm noticing that there are several of them, but there's space in between, almost as though they're floating around. So I would put my observations in here for the bacteria cell, and then we move on to the next section. This is a chance for you to self-check. I'm going to pause and let you answer these questions, and then we'll meet back at the other side. Which of the specimens that you observed are made of cells? All right, so the first portion is complete. Now we move on to cells come from existing cells. In this experiment, you will follow in the steps of great Louis Pasteur. See if you can find support for the following hypothesis. Cells only come from existing cells. Record your observations in your lab report. So here we have Pasteur's setup. He set out to discover if cells could come from spontaneous generation, meaning that they could come from non-living materials such as air or water, or if they only came from other living cells. He filled 2 flasks with broth and heated them to kill any existing microorganisms. Repeat his experiment with the flasks below, which contain identical samples of a broth with nutrients. Begin by heating up the flasks. So we're going to select the knob here to heat it up. Now we're going to cool them so you can see they're burning. Now we're going to cool them down. Select the neck of this flask to break it off. So this flask is open to the air, whereas this flask doesn't have as much access to the air. This is day 0. Pasteur observed what happened in each flask. We're going to repeat his work by observing what happens in each flask over 5 days. Record your observations in your lab report. So looking at the lab report, you have day 1. You have the flask with the broken neck that's open, and the flask with the intact neck. Write your observations for each. Same for day 2, day 3, day 4, and day 5. So if I move on to day 1, what do I see here? Color, shape. Make sure this is the one with the, they call it a broken neck. I just call it, that sounds really bad. It's just an open neck. It's not broken. This is the closed one. Here's day 2. What do you see? What colors are you observing? What shapes? Number 3, look at the colors. Number 4, what do you observe? And day 5, what do you observe? Also notice things here are happening. That was happening. Look, I can go back and look. I can see things are forming here. Day 1, day 2, day 3, day 4, day 5. Once you've recorded all of your observations for both flasks, you're going to move on to the next part. Here's the self-check. I will meet you on the other side. All right, third experiment. Cells are the basic unit of life. What makes something alive? Check all that apply. I will meet you after I've finished mine. So in order to be considered alive, a thing must grow, reproduce, and use energy and nutrients. Part of cell theory states that cells are the basic unit of life. How can we test this? In this experiment, you will attempt to grow colonies of bacteria cells under 2 conditions. One will provide energy and nutrients for the cells, and one will not. What will happen? Record your prediction in your lab report. Scrolling down here, they don't have the spot for the prediction. Interesting. I don't believe it's up here either. Nope. Nope. And I'm not going to add it. We're going to leave it the way it is. There we go. Let's go back. All right. Petri dishes are small circular plates used in labs. The agar solution is a gel-like substance used to grow small samples of bacteria. Add agar solution to each Petri dish on the lab table. Begin by dragging the solution with nutrients to the Petri dish and then select Next. There. Now we're going to drag the swab to the bacteria to pick it up, and then we're going to apply it to the Petri dish. I go back here. There we go. And we have day 0. Day 1. Now we have the agar solution with nutrients, and this is the solution without nutrients. So notice what's happening. Size, shape, amount. Day 2, day 3, day 4, day 5. You will record what you observe in this table right here. Then you come to your conclusion questions. When you're finished here, you're going to be sure to come and answer these 3 questions in the space below, and you have finished your lab report. Yes, this is a long video, but I've walked you through exactly what you need to do. I look forward to seeing your work. If you have any questions or concerns, know that you can always reach out by email, phone call. Leave me a message if you do call, because otherwise I don't know you called. Or you can book an appointment with me and I'm happy to go over this with you. Good luck. Bye, guys.